Hi, my name is Kate Broadbent. I'm the Associate Director of Bioinformatics Infectious Disease at Illumina. And we'll be sharing today a app demo for the RPIP Respiratory Pathogen ID and AMR panel app that is live in BaseSpace. And quickly want to cover the fact that this app is a new Illumina app that replaces a kind of prior app for RPIP. So if you search here in the Illumina BaseSpace app, you are going to see two applications available. This Explify RPIP Analysis with the ID by DNA logo is kind of the app that's being replaced by this more latest Explify RPIP Data Analysis app that is Illumina branded and has a lot of new features, but very similar bioinformatic results. So we're going to jump right in here to the Explify RPIP Data Analysis application. So the first thing you'll see here is a description of the application. I think this is really important to go through carefully and understand the high-level features that this application provides to users in a kind of end-to-end fashion. It is a tailored data analysis solution for a set of probes, an enrichment kit that you can read more about and click on this link here in the app description to understand how to order that enrichment panel kit. A, B, and C, and then a lot more product highlights and information and content is available through this website here in terms of application notes and descriptions of every kind of organism, AMR marker, drug class that is covered by this panel. Another important thing to note here is that we've provided a demo project that can be imported by clicking on this link or searching in the demo data section. So if you go into the demo data section of BaseSpace, going a little slow apparently. Not sure why this isn't. Demo data, you can see this project right here, Explify Respiratory Pathogen IDM AMR Panel RPIP Demo Project. You can read a bit about what is the samples we've included here. So there's 4 samples. They include a quantitative internal control, which is the one that we recommend and have tested the most at Illumina here, which is the phage T7 control. It's at a recommended concentration, spike concentration of 1.21 times 10 to the 7th copies per mil of sample. Also, 3 out of these samples have been spiked with a human background. So you'll see that kind of human read contribution coming through. It's important to remember that this is an enrichment product, not a full subtraction product. So you can still see some human reads coming through, even though the probes are not enriching human sequence. These samples were processed on a NextSeq with the enrichment panel kit at 1 by 147 base pair sequencing length. And we do recommend 1 by 150 or 1 by 100 as the recommended read length for these products. You can see that this data has been analyzed with default settings, and we'll jump into what the non-default settings are back at the app interface. Okay, so at a high level, we're providing broad identification and quantification of respiratory pathogens, SARS-CoV-2, influenza viruses, RSV virus, hard to culture or long culturing organisms like Mycobacterium tuberculosis and Legionella species. We're providing insights on antimicrobial resistance for 26 relevant drug classes. We're providing strain typing of critical pathogens, so full genome consensus sequence for SARS-CoV-2 and influenza AV viruses, integrated pangolin SARS-CoV-2 lineage assignment. We're providing many quality control features, so you can elect to use automated read QC, to include a internal control. So one of 10 commercially available spike-ins can be used as a process control as well as an absolute quantification control. We're providing sample composition profiling, which provides an indication of how much host versus microbial reads are both targeted and untargeted in the final sequencing data. And a lot of different flexible analysis options and result formats that we'll look at more closely. But at a high level, there's a summary PDF report. This kind of gives you a first page quick look at the most important results in a sample. There's also a convenience Excel report that summarizes a lot of information that's provided in this comprehensive JSON report that really just covers everything in the sample, from QC metrics to sample composition to microorganism detection, AMR detection, variant detection, an interactive HTML web report, which we'll also take a look at, annotated variant tables for SARS-CoV-2 and 3 of the drug resistance related segments for influenza A, as well as any known bacterial AMR marker variants conferring antimicrobial resistance. And then, as I mentioned earlier, consensus genome FASTA sequence for SARS-CoV-2, influenza A, and influenza B. So that's all 8 segments, full genome for those 2 segmented viruses there, influenza A and B. And also show this additional aggregate Excel report feature that users can elect to download if they are interested in getting a holistic summary of every sample in a project. Just quickly of what else is here on this app landing page. This again is replacing the previously available Explify RPIP Analysis app in base space as issued by ID by DNA version 1.0.1. This is now version 2.0.0. We've got a section of known limitations. I think an important one is to, definitely take a look through all of this. I think an important one to be aware of is that there is no bacterial AMR markers reported for read lengths below 60 base pairs. So we do want to make that aware. And then we have technical release notes, important to track your versions and kind of what's changing in terms of high level, what could change for a customer in terms of like actual microorganism or AMR marker reporting. We have the latest Pangolin version, 4.1.3 released with this RPIP product. And we've also made an update to the carbapenemase flag for AMR markers and organisms, where that is now flagged at a drug class level rather than at an antimicrobial level. And we'll get more into these as we look at the app interface in terms of the read QC and other configurable options, some minor changes to the PDF report file and JSON report file. Okay, so after you've looked through that, looked through the EULA and the privacy policy, and you're ready to launch your application, version 2.0.0. We can take a look here. So you can launch a analysis here in base space with either a project or a biosample list. And what this app is assuming the input to be is demultiplexed and adapter trimmed FASTQ files. And that can be selected here as a project or a list of biosamples to analyze. So pretty standard input. Then here are the options. So the defaults that are automatically kind of defaulted to mirror what was available in the 1.0.1 application, where read QC, so low quality read trimming and filtering, is performed. AMR markers are reported only when an associated microorganism is detected. That's by default. The absolute quantification is performed using Enterobacteria phage-T7 at this recommended concentration of 1.21 times 10 to the 7th copies per mil of sample. And we're going to report all microorganisms, all 282 pathogens that are targeted by the RPIP probe set. So that's, again, the default. But what we've made flexible and configurable is if you want to customize this a bit further. for your own sequencing data. So if you've already performed read QC, or do not want to use our automated read QC, you can turn this off. You can uncheck this box. This little pop-up here is also going to give you a little more information on what the toggle buttons are going to do for you. So again, if checked, reads are going to be pre-processed using quality metrics before analysis. If they're unchecked, we're still going to calculate the quality metrics and output that, but no reads are going to actually be changed prior to the Explify analysis. This other toggle button here, or toggle checkbox, if checked, again, that's the default configuration. Detected AMR markers are going to be reported if the AMR marker passes minimum analytical detection thresholds, but also use some logic to be sure that one or more associated microorganisms are also detected and reported. As a quick example, the MEC-A marker is associated with Staphylococcus aureus. If there was a sample in which MEC-A and no Staphylococcus aureus was detected, with this button checked, you're not going to see that MEC-A marker detection analytically because the Staph aureus was not detected. But if you uncheck it, you can basically bypass that additional logic and report your AMR markers as long as they pass an analytical detection threshold without any relationship or linkage logic to the organism side of reporting. So possibly kind of a surveillance use case. In terms of the quantitative internal control, we now support a user selecting any of the 10 commercially available and targeted internal controls that are included in the RPIP probe set, as well as none. So if you're not going to use a quantitative internal control, don't want those results in your report, we don't recommend that and think there's a lot of added value, certainly, with having that internal control be a quantitative internal control and not just a process control, and having that additional information. But if you do select none, it's going to gray out this concentration and this field will be ignored. But if you want to change, if you want to use T7 at a different concentration, you can, edit that here. If you want to lower it by a log, you can edit that here. If you want to use a different internal control, RNA phage, MS2, for example, you can select that here, and Explify will perform the absolute quantification calculations with that alternative internal control. And again, you can change this concentration. There's some guidelines on how that needs to be input in terms of format. If you happen to, as getting used to this, input a incorrect format, the app is going to immediately error and not let you submit the job. It's going to kind of do a check on this format of this free text field here. So some good examples, 1 digit left of the decimal. So yeah, 1 digit left of the decimal. So this is some valid examples. You can see that this is not valid because there's 2 digits left of the decimal. You can see right there. And in this case, there's 0 digits left of the decimal. So hopefully that's easy to get used to. Once you've done it once, you can do it again. Last kind of configuration option here is the microorganism reporting list. So again, default will report all 282 pathogens across the bacterial, viral, and fungal spectrum. However, if you're only interested in a subset, this is kind of a virtual filter on the results, if you will, to, for example, only report respiratory viruses, only report fungal microorganisms, or these other kind of pre-selected, predefined sets of organism lists. So there's 5 pre-selected organisms lists. There's also, if you're just not interested in pathogens or microorganisms and only want to report antimicrobial resistance markers, only see AMR detection results, you can also select this, and that's going to automatically unselect your marker, your checkbox here, if that makes sense. Such that you can report those AMR markers in a AMR marker only fashion without any connection to the associated organisms. So I'm going to switch my screen quickly. There's a panel summary that is provided with RPIP through the web page that gives a high-level overview, but also a detailed overview of everything that's in a panel. And I just want to show that we have basically, spelled out exactly what those microorganisms subprofiles look like. Hopefully I'm choosing the right. Yep. So here we have an executive summary tab that gives a high-level summarization of how many microorganisms, how many AMR markers are covered in the full default analysis. I just quickly wanted to show that there's also this list here that provides those 5 subprofiles, and so you can take a look at whether one of these is a relevant subset of pathogens that you would want to potentially virtually filter your results to. Okay, so I'll jump back to base space screen. Where were we? Okay, so now we're going to look at some results. So we have launched our results. We're going to take a look at the demo project data. So that is here. And in here in this demo project, you have the actual FASTQ files. So those are those have been uploaded here, these 4 upload events. And then you also have the analysis results to start looking through. So you can see the FASTQs are found here in the FASTQ section. And then we're going to jump into this analysis folder here. I do want to highlight that for support tickets, one of the important fields to capture as a user or as a support, if you're going to send, if there's a question about the results, capturing this analysis ID right here, it's on the analysis page, the 6213-00792 is going to be a really important piece of information to provide if you're submitting a ticket through the SET platform for any sort of question related to a result or sample to look into more closely. Just wanted to highlight that there. But what we're seeing here is our report section where there's a high-level report showing how many total raw reads, unique reads, % unique reads, and post-quality reads there are for these 4 samples that are in our demo project. We have 4 samples in here. One has Klebsiella pneumoniae, one has RSV, a relevant virus. One is a polymicrobial sample that we can look into that has bacteria and virus, SARS-CoV-2, and then one is a negative control with no pathogens detected at all. They all have roughly our recommended total raw read recommendation of approximately 1 million reads. So these are just a little bit under that, but quite close to the 1 million read benchmark. And these have all been spiked, as I mentioned earlier, with a quantitative internal controls to provide quantitative results. There is this button you see right here to download a combined Excel file for all samples. All you do is click on that and it'll download the sample. I don't think the pop-up is showing, but we'll take a look at that combined Excel after looking through the individual sample results. So this is the summary report. There's also a report per sample that I'm gonna quickly show. So we'll jump down into this sample. This is the polymicrobial sample, probably kind of the most interesting. So we'll look at this one. We have here a kind of semi-interactive web report where you can do some toggles, you can do some hover buttons to look at what the legend for the sample composition graph means. And you're also looking at your internal control. So these are the 10 internal controls. This is the RPKM for the quantitative internal control that was used in this experiment. Also some additional QC metrics, your mean read length is important to keep an eye on. Total raw reads, important to keep an eye on. And then you've got more kind of pathogen and AMR specific results that you can look through to just get an early gauge. So SARS-CoV-2, you can take a look at the definitions of phenotypic group. There's a pop-up, not sure if that comes through in the video, but gives you the definitions of the different groups. You can see here, you know, high coverage, high average nucleotide identity, relatively low median depth and RPKM, but, you know, present at 1 × 10⁵ copies per mil or thereabouts. We have 2 bacteria also in the sample, Klebsiella pneumonia at quite a high level, and you can see some correlation there between your quantification and your RPKM and median depth, indicating that pathogen is at quite a high load. There's also intrinsic resistance provided for bacterial pathogens at a drug class level, and information on AMR markers that have been detected that have a associated organism that was also detected, because this was run under the default analysis options. This can also scroll over to see the full list of associated drug classes. Okay. Also going to show in the files section here what you can see for the same sample. So this is this polymicrobial sample. We've got multiple different result formats. So I mentioned this PDF file. You can get a small preview here, but this is kind of a branded, nice looking report that gives similar information, just presented in a PDF format. We can take a closer look at that I'll pull one up in just a moment. There's also a JSON report here that's very comprehensive. There's this new result format, an Excel file, and then there's the consensus genome FASTA provided here for SARS-CoV-2. If influenza had been detected, influenza A or B, that would also be generating a full genome FASTA for the 8 segments there. And then this is specifically pulling out the variants detected in this case in SARS-CoV-2 or known bacterial AMR markers. So I'm going to share a little closer look, quick preview of the PDF and JSON and Excel format. And head over to that. So, okay. So I'm going to share my screen again. Okay, hopefully you can see this now. So this is a PDF report where some of our most important results are kind of shared on what we call the first page, even if it runs onto a second page here. I mentioned this earlier, I wanted to highlight again that this potential carbapenemase and carbapenemase flag has changed, has expanded. There's just additional flags that have been added to markers to flag that at a drug class level rather than a individual antimicrobial level. And we're providing drug class level information in the PDF report now. But that's, I guess, basically kind of what this looks like on the first page. Of course, scrolling around here, sorry, the quantitative information is provided, relative abundance within the bacterial kingdom, and then 100% because SARS-CoV-2 is the only virus, that's that relative abundance, that proportion, indication of whether an associated AMR marker was detected in that phenotypic group. And this is kind of like a biothreat agent classification. So 3 is your least likely to be commensal, most likely to be a true disease-causing respiratory pathogen. And then there's also additional information provided in additional tables, high-level reclassification results. So just quickly at a glance, there's an antibiotic drug class table. There is also anti-tubercular and antiviral drug class table, if those are relevant, and then this commonly associated AMR marker and microorganism table. Okay, I'm going to jump over to quickly show the JSON report. And yeah, pull that up real quick. And then we'll finish on the Excel format. Okay, so this is a JSON report. It's a kind of structured schema with defined fields and data types, and is, you know, comprehensive. Everything in the PDF report and the Excel formats, the more convenient formats are also provided in this, you know, comprehensive machine readable JSON report that we envision to be what's consumed by, you know, potential LIMS systems in a scalable manner. But all the information is there and more. So, internal control results, the microorganism results, AMR results, etc. Sample composition results. So everything is provided in this JSON report. There is a analysis app user guide that provides a definition for each of these fields. And these map quite intuitively, we hope, to the Excel format that we'll pull up next. Okay, so pull that up. Okay, let's see. I think I need to download that one. Oh, here we go. Sorry. Here it comes. Okay. Screen. Okay, so hopefully I got this. So this is the file name that the application is going to give this. It is going to be kind of whatever that analysis was named as, and it contains the analysis ID, which is, again, that important value if you want to submit a ticket for support and for the bioinformatic team or others to go look into the results if there's any concerns or questions. So that analysis ID, you just want to highlight, is an important kind of hook into the results. This file, though, is organized as a samples tab, a microorganisms tab, an AMR tab, and a variants tab. And what's really convenient about it is that it's, again, this this aggregate format for all 4 samples that were in this project. And here in the samples tab, you have kind of high-level information about what your detected targets were, detected microorganisms, detected AMR, kind of whichever way you want to have that combined or split out. A lot of your QC information, your information on read length, before and after quality trimming, so pre and post quality mean read length. And then you've got your high level 4 classes of targeted, untargeted, ambiguous, and unclassified reads. And then you've got your breakdown within targeted and untargeted into the different targeted subcategories and your untargeted subcategories. And that first one is a relative within each, you know, these add up to 100%, if that makes sense. And these add up to 100% right here, all the way to there. And then these are your absolute numbers for those same values across the whole set of data. And then we're moving into, one more right there. And then we're moving into the 10 internal controls, looking at the RPKMs across those 10 ICs, which one has been used for quantitative reporting and what the input concentration was. This is always important to track the versions of the analysis pipeline and the test version. So this is 5.11.3 for RPIP. And then what time the results were generated at. So for the microorganism tab, this is kind of 1 row per microorganism result. And so for this polymicrobial sample, for example, you're seeing 3 different rows here for the 3 different microorganisms that were detected in this sample. And all the kind of important classic bioinformatic metrics are shown here: coverage, ANI, medium depth, and RPKM. And then additional information, if relevant, pangolin, lineage, and associated AMR intrinsic resistance or drug class resistance. Makes sense. And then we've got an AMR tab, which again is one row per AMR detection result. So we'll just take this, actually let's just take this first sample, this K-pneumo sample. These are the AMR markers associated with this K-pneumo detection. And again, similar sorts of lots of content here, card bit scores, threshold, and bit score that was detected in the sample, amino acid, protein alignment metrics here for coverage, PID, median depth, and RPKM, and a lot of information on the associated drug classes and provided here. And then the last table is information on variants. So in this sample, we're looking just at the SARS-CoV-2 result as well as the polymicrobial. Yeah, the SARS-CoV-2 looks like it's driving what's provided here. There's not any bacterial variant markers or influenza known variant markers. And so these are just de novo viral variants that are annotated here in terms of whether they're synonymous or non-synonymous, frame shift, et cetera, in this sequencing result, with also their depth and allele frequency. Okay, I think that is what I wanted to share. There's also a UPIP very similar app, but we'll touch on that at another time.